Okay, so we're doing the light-dependent reactions. Let's just start off with the general equation of photosynthesis. So here we're going to see CO2 plus, and I'm going to put in a different color all of the things that come into play on the light-dependent reactions. So in this reaction, we will see the water come into play. We will see the solar or light energy come into play. And that is going to go to a sugar, which is not generated in this reaction. So we will not see that sugar today. No, it's just in black. So I put the things in black that are going to be used in the next reaction. So carbon dioxide, we're not dealing with that today. Today we're dealing with water, solar energy, and oxygen. That's about it. Okay, so that's what we're looking at. All right. So if we were to come into our diagram here, there's some stuff that I want to label initially before we actually get started. So here, this protein right here that's in the membrane, this is going to be photosystem 2. So I'm just going to do PS2. Okay. And then this one over here, what would this be? Yep, that'd be PS1. Okay. So we have photosystem 2, photosystem 1. Remember that photosystem 2 actually acts first. Photosystem 1 is the second actor in this whole process, but they were discovered in the opposite order. So PS1, photosystem 1 was discovered first, photosystem 2 is discovered second, and that's why they were named that way. It has nothing to do with the order that they actually act. I know, it is kind of annoying, but it is what it is. Okay, and then we have a series of proteins here, and it only shows 3 of them. We don't need to know any of their names, but all you need to know is that from here to here, we are dealing with an electron transport chain. So that's all part of the electron transport chain, from here to here. Okay. All right, let's also label where we're at. So we are in here in the thylakoid space. Which means that out here would be the stroma on both sides. Okay. So again, if we were looking at this, we're in a membrane, and then on the outside we'd be in the stroma, which is just like the cytoplasm of a chloroplast. Okay. The thylakoid space is going to have a high hydrogen ion concentration. The stroma is going to have a low hydrogen ion concentration, so low H+. This membrane here is the thylakoid membrane. Notice that it is set up a lot like our cell membranes, so it's just phospholipid bilayer all the way around with some embedded proteins. Okay. And I think that is the only thing we need to label for now. Okay, so let's get started with the actual stuff. So the first thing that we're going to see happen in the light-dependent reactions, so what I'm going to do is I'm going to number the actual verbiage, and then we will color in and number where that's happening on the diagram, so you can reference back and forth. So the first thing we're going to see is that photons, which is just light, okay, we're going to strike the chlorophyll. Within photosystem 1. Or no, I'm sorry, photosystem 2. So strikes the chlorophyll within photosystem 2. That's going to be our first actual step. And then that's going to cause some things. So I'm going to put bullet points of what that causes. So every time I put E minus, What does E minus mean? Electrons. Okay, so electrons, okay, W/IN just means within. If you didn't want to, I'm using some freehand just to shorten this up. Within the chlorophyll are excited. And move to the primary electron acceptor. I like how they call it that. Primary. It has a name, you know. Electron acceptor. Anyone get curious and Google search what the primary electron acceptor is actually called? I guess I'm the only big enough nerd to do that. It is actually called the pheophyton. So it's a pheophyton. I don't know. It's a weird name. Go ahead and put it in there. That it kind of does. Pheophyton. Okay. So the primary electron acceptor, pheophyton. Okay. So that's going to basically move it up into kind of this loaded position where the electrons that were in the chlorophyll now have bumped up in energy level, so now they're high energy electrons. Okay. And then what else is going to happen is photosystem 2, so PS2 here, uses energy from that light, uses light energy to catalyze, means speed up. So photosystem 2 is a protein and enzyme in one. It's this really massive complex protein that has an active site on it that splits water. So catalyze the splitting of water. So we're splitting apart a water molecule, and this is what gets released. So we're going to get out of this. Oxygen is going to be released from the thylakoid. Okay. We are also going to see 2 hydrogen ions for every 1 water molecule. They're going to get released and remain in the stroma. Or no, not in the stroma. Sorry, they remain in the thylakoid space. There's an error on what I wrote yesterday. Okay. And then 2 electrons replace the electrons that are now at the, instead of writing primary electron acceptor, I'm just going to do PER. So primary electron receptor. Okay. So let's go in and actually show what that would be. Or no, it's acceptor. A, PEA. So it's a P. Okay, so let's go ahead and put all of that onto our diagram here. So I'm going to move over. So process number 1 is happening here. We're going to have light energy come in, and I'm going to do all of this in the same color that I wrote the stuff in. This way you can very quickly cross-reference the verbiage that you wrote with where it's happening on the diagram. So I wrote it in red, I'm filling this in red. So light comes in. And then what we're going to see happening here is these electrons are going to get bumped. So here are the electrons that were contained within the chlorophyll. They're going to get bumped up to that primary electron acceptor, and we should actually label that. So it's this little box they put in there that's representing it. And then we're going to see water get split. And these electrons from the water are going to go back in and refill that position. And basically, this is just a way of every time that light comes up and kicks out electrons, there's new electrons that are able to come in from the breakdown of this water. So this water being split is also part of this first process. Okay. And then we said that there was some stuff released. So there's the electrons that got released. They go right back in to photosystem 2 to replace the ones that are now up here with the primary electron acceptor. Okay. We're also going to see these hydrogen ions get released. So we have these hydrogen ions here. And then we had some oxygen get formed, and the oxygen is going to be released outside of the thylakoid membrane into the stroma. Why does this oxygen not have to go through a protein to get out? Because it is small and nonpolar. Exactly. It can diffuse. Okay. So it leaves. So that would be all of the first step stuff. Okay. Next. So now here's what we have. We have two things that can be used. We're going to start using these hydrogen ions to make ATP, and we're going to use these electrons up here to make NADPH, which are the two main goals of this whole process. NADPH is going to happen across the top. ATP synthesis will happen across the bottom. All right. Part 2. So electrons from the P, primary electron acceptor, enter ETC, the electron transport chain. And as they move through the electron transport chain, just like we saw in cellular respiration, they're going to be releasing energy every time they move down a level. So, yes. Releasing their stored energy. As they interact. With the proteins. Proteins of the ETC. Okay. And then eventually they return to their ground state. What does it mean for an electron to return to its ground state? Ooh. This is a chemistry question. So an electron at its ground state means that it's at its lowest energy level. It's at its least excited. It is at its most stable location, lowest orbital. Okay. So essentially these electrons started off really high energy. They're going to start moving through the electron transport chain, and every time they do, they start releasing some energy that's going to be used to do something. And here's what it's going to be used to do. So here are the results of this occurring. So the negative delta G, what does that mean? So the released energy, the free energy from the electrons are used to do something exactly like what we saw in cellular respiration. So what did the electron transport chain do in cellular respiration? The energy was used to pump hydrogen ions. Yeah, same thing here. So are used to pump hydrogen ions from stroma, so from outside of the thylakoid to the inside, into the thylakoid space. Okay, and this is going to set up a hydrogen ion concentration. So it sets up an H+ concentration. gradient. So this sets up a situation where now we have a higher concentration of hydrogen ions inside of the thylakoid space than outside. And now that can use chemiosmosis to perform an actual process. So then the next thing that we see from this same situation is that electrons from the electron transport chain, ETC, move into PS1. So they're going to move from the electron transport chain into photosystem 1. Okay, so let's look at what's actually happening then in the diagram. Are you good here? Do I need to slow down? Barely slow down? All right, so here's what's going on then. If we move over here, we just had electrons move up into the primary electron acceptor in our first step. Now we're going to see them flow through the electron transport chain. So I'm just going to color in these arrows to show that this is where it's headed. There, and then there. And then finally here is where it enters photosystem 1. Okay. And again, this is all within the electron transport chain. Meanwhile, the whole time that this is happening, so this is step 2 here and step 2 down here. So I'll just put it there. The whole time this is happening, every time that the electrons move into a new protein within the electron transport chain, we're going to be taking hydrogen ions that are out here and pumping them in. So like if I had a hydrogen ion right here, it is going to come in and follow this arrow. And enter. And now here's my hydrogen ions that I just pumped in. So the role of the electron transport chain is to pump hydrogen ions from outside in the stroma to inside the thylakoid space. That's the goal. What's that? Like, why do they not eventually run out? So the Calvin cycle is going to release these hydrogen ions into the stroma. Yeah. So it's a recycled process. Yep. Okay, so we're getting hydrogen ions from two places then, from splitting water and also pumping them into in from the stroma. Okay. So step 3 then. This is a 4-step process. Well, I broke them into 4 steps. Okay. Hydrogen ions diffuse out. Can hydrogen ions go through the plasma membrane? Like, can it go through the phospholipid bilayer? No. Has to go through a membrane protein. So this sets up a one-way exit for these hydrogen ions. Hydrogen ions diffuse out of the thylakoid space. Into the stroma. Through ATP synthase. And what this does is it synthesizes ATP. Through the reaction of ADP plus P to ATP. Oxidative phosphorylation. Gotta love it. Okay. So that is the end of step 3, so we can add that one to the diagram now. You guys good here? Give me a minute. So the electron transport chain and the splitting of water set up this concentration gradient, and now we're using that concentration gradient. We're good? So go on in now. We're going to start using this. So step 3 is happening here and here. Okay. These hydrogen ions are now going to come in from their 2 locations. They're going to find their one exit out because they want to leave. There's so many more of them inside than outside because remember the thylakoid space has a really high hydrogen ion concentration. The stroma has a low hydrogen ion concentration, so they're trying to get out. And this is their only way to get out, is out through this ATP synthase, which I forgot to label. ATP synthase is the name of this enzyme. And it is going to be taking ADP plus P and making ATP. Okay. Almost there. So we have one of our main goals done. ATP is made. So the other thing that needs to be made is NADPH, which is the electron carrier. Because in order to make glucose, we need 2 things. We need energy from ATP, and we need free electrons from the NADPH. Okay, so step 4. Okay. Oh, really? Oh, wow. Okay, fair enough. Well, yeah, if you need to like flip to the back or grab another piece of paper, we can do it. This one's not long, but I have another thing to add after it. Okay. We got this. It's okay. We're learning. So electrons from photosystem 1 now. So the last where we left off up here was we had electrons leaving the electron transport chain and entering photosystem 1. These are entering at a really low energy level. We need to excite them again. And that's going to be the role of light hitting photosystem 1. So electrons from photosystem 1 are energized. By incoming light. And used to reduce NADP plus to NADPH. So we reduced it, meaning that we gave it electrons. So these electrons were energized and then used to reduce the NADP plus. Okay, and this is done by the enzyme NADP plus reductase, the most literally named enzyme I've ever heard of in my entire life. Okay. Yeah, so that's step 4. Okay, so we can show that happening now up here. Give you guys a chance to finish on that one. Reductase, yeah. It's labeled on your diagrams up there. All right, so this is happening right here. We have incoming light. This is step 4. It is going to bump these electrons that came in up another energy level. And then they're going to flow through another little series of proteins that we don't need to know. And then finally that energy is used by ATP, or by NADP reductase to take incoming NADP plus and spit out NADPH. So it was reduced. Both of these products, the ATP and the NADPH, are going to go to the Calvin cycle. So now we have our two final products. The last couple things I want to add in is just like kind of a brief summary of what's going on here. So I'm going to use my two colors that I used for step 3 and 4. So those are my attempts at asterisks. ATP, and I'm doing the colors that we made them in. NADPH. Produced in chelate reactions move into the Calvin cycle. Where the energy from the ATP and free electrons from the NADPH are used to reduce atmospheric CO2 into, so I'm going to put biologically significant sugars. You can just say sugars as long as you realize that some sugars can be used in the Calvin cycle, or some sugars can be used in cellular respiration and some can't. Like G3P cannot be directly used, it has to be converted. So biologically significant sugars, such as like glucose and fructose, and etc. Others. Sucrose and maltose and galactose, yes. Reactions. I'm just shortening reactions. Frictions. It works, right? Okay. So, if we were to zoom out on this whole process, we used light energy to split water, to excite electrons, and to produce hydrogen ions. The hydrogen ions go on to make ATP. The excited electrons are going to move down, pumping in more hydrogen to make ATP, and then get re-excited in photosystem 1 to make NADPH. I feel like this process gets made a lot harder than it is, a lot like cellular respiration does. I'm hoping that this kind of like helps to narrow your focus on what's actually happening. The last thing I want to do is just fill in this little guy that's going to link both the light reactions and the Calvin cycle. So this is the light reactions that we just went over here today. We would have incoming water and photons, so light energy. We would be producing as a byproduct oxygen. As a main product that's going to be used, we make ATP, right? And also NADPH, which is just an electron carrier. Right. And this occurs in the thylakoids. The second half of photosynthesis takes place in the stroma, and this would be the Calvin cycle. And in the Calvin cycle, what we'll see is it's going to be incoming CO2 from the atmosphere is going to come in, and we'll perform a series of reactions using the ATP for energy and ATPH for its electrons. We are going to produce sugars, and the now oxidized ATP and NADPH are going to leave as a DP plus P and NADP plus. So we have now opened up more NADP plus to perform light reactions again, and the whole thing just keeps on repeating over and over and over again. And again, the plants are going to be producing more oxygen than they consume, which is going to oxygenate our atmosphere. So again, the products of cellular respiration form the reactants of photosynthesis and vice versa. And this is occurring in the stroma. All right. 4 questions. What do you think? Go ahead and end this real fast.